All right, we're going to pick up where we left off on this is section 6, chapter 9, section 6. So in the event of we don't get to finish in class. So glycolysis, right? We started out by saying our very first step was hexokinase. And hexokinase is that very first irreversible reaction. And it's hexokinase or glucokinase, which we talked about in class, right? And all we're doing here is we're adding a phosphate group. So what used to be a hydroxyl is now a phosphate. Where the phosphate come from ATP. Okay. So our second one is phosphoglucosisomerase. So think about what an isomerase does. It's just an isomerization. We're just going to kind of rearrange some things, right? So we had originally a 6-membered ring in glucose, right? And now we're going to convert that ring from being a 6-membered ring to being a 5-membered ring, with the 6 carbon now being outside of the ring instead of inside of the ring. And so we go from being glucose 6-phosphate to now being fructose 6-phosphate. So that's the difference between glucose and fructose, right? One is an aldose, one is a ketose, right? Has an aldehyde or has a ketone. All right, so that's just kind of a rearrangement. And now what you're going to see is kind of more symmetry to the molecule. And so now we have a carbon over here that can then be phosphorylated, which is the whole point of the next one, which is phosphofructokinase. So kinase tells me that I'm going to attach a phosphate group. So we had a phosphate group on the carbon on this side. Now we're going to put a phosphate group on the carbon on that side. So that's exactly what you see. We convert this hydroxyl into a phosphate group. And so this is our second investment, right? We had our first investment where we had ATP, where we put in, ATP. So this is our first investment. And then this is our second investment. So this is where we're putting in those ATPs, right? You got to spend money to get money. So this is our Phosphofructokinase PFK-1. So sometimes you'll see it abbreviated that way. But this is the rate limiting step of the entire glycolysis pathway. And so what you're going to see is that this can be regulated. This enzyme, phosphofructokinase-1, can be inhibited by this can be inhibited by ATP. And that makes sense, right? So if you have a lot of ATP sitting around in the cell, do you want this to continue? Well, no, not really, right? What if you have not a lot of ATP? What if you have AMP or ADP? So this is going to have the opposite effect. It's going to stimulated by high levels of AMP and ADP, right? So these, the ATP, the AMP, the ADP, these are all allosteric regulators of phosphofructokinase-1. And you got to think, do you want glycolysis to continue? Yes or no? And that depends on the current ATP status of the cell. And so this is our other irreversible reaction. But this is really the rate limiting step of the entire pathway. So you're going to convert from fructose 6-phosphate to fructose 1,6 bisphosphate. And so that's what the BP is, bisphosphate. And the bisphosphate is just telling you that you have phosphates attached to 2 different carbons instead of 2 phosphates attached in sequence. Like ATP, adenosine triphosphate, that'll be 3, right? Bisphosphate is telling you that they're on 2 different carbons. Okay, so the next reaction is an aldolase. An aldolase is going to give us a cleavage event. So this is what we want, right? We want to get to the point where we go from having a 6-carbon ring into having 2 3-carbon chains. And so that's what's going to happen. And this cleavage is going to be between our C3 and C4 carbons. Now, what you have to remember, and your book doesn't show this, is that this ring structure is in an equilibrium with the chain structure. And so you actually have this aldolase reacting with the chain instead of with the cyclic molecule. And so it looks like you need 2 cleavage events, but you don't, you only need 1 because this ring is in equilibrium with the straight chain. And so what ends up happening is you actually get 2 different products. You get Glyceraldehyde-3-phosphate, which is great. This is an aldose. That's awesome because Glyceraldehyde-3-phosphate is a substrate in the next reaction. But we have a problem. We also made dihydroxyacetone phosphate, and we can't use it. That's a ketose. So what we're going to have to do is we're going to have to go ahead and get this to be converted over to Glyceraldehyde-3-phosphate in order to proceed, right? So how are we going to do that? We're going to use an isomerase. So this is a Triose Phosphate Isomerase, right? So you're going to use those 2 dihydroxyacetone phosphates, or DHAP, in order to convert it into Glyceraldehyde-3-phosphate. So this is the end of our investment stage. So this is the end of basically part 1. And what you can think about, what have we done so far? We started out with 1 glucose molecule, and we have ended up with 2 3-carbon chains, right? 2 G3Ps, so glyceraldehyde-3-phosphate, that's what we really want. And we've invested 2 ATP. So now we're going to make some money. We invested some money, we're going to make some money. So this is our stage 2. Stage 2. And stage 2 really has some key features inside of it. So inside of stage 2, right, we have those 2 glucose 6-phosphate glucose 3-phosphate molecules per that 1 glucose, right? So we have this glyceraldehyde-3-phosphate right here. We have really 2 of these. So it's really times 2, okay, for every 1 glucose molecule. And so now, instead of taking a phosphate away from an ATP, what we're going to be doing is we're going to be giving that phosphate to an ADP and making an ATP. So what we do is we call this substrate-level phosphorylation reactions. It means the phosphate group is going to come from the substrate. And so this is going to generate us 4 ATP, but we know we already spent 2. So this is how we're going to get our net of 2, right? Okay, so, and the other thing that's important is that at the end of this, we're going to need an irreversible step so that all of the pyruvate that we make stays as pyruvate and doesn't go backwards in the reaction. So, if you have your glyceraldehyde-3-phosphate and we have an inorganic phosphate, what are we going to do is we're going to slip on an extra phosphate on the other end of our glyceraldehyde-3-phosphate. Now we have 2 phosphates, 1 on carbon 1, 1 on carbon 3, and so we call this new molecule 1,3-bis-phosphoglycerate. And that was catalyzed by glyceraldehyde-3-phosphate dehydrogenase because it was a dehydration reaction that allowed this to continue, right? So a dehydration is telling us that we're going to take a hydrogen and a hydroxyl, right? And we're going to make water with that. And then this phosphate is going to go in and attack that carbon, right? And so that's how we get that addition of our inorganic phosphate group onto our glyceraldehyde-3-phosphate. Okay, so now we've got a phosphate group on either end. Now what we want to do is we want to use that. So the really cool thing about our product from our previous reaction right here, this guy, 1,3-bis-phosphoglycerate, this is super high energy. High energy, so this one. They're the same molecule. These are high energy, right? And so remember that we actually have 2 of them. So we're going to make 1 ATP because we're going to transfer this phosphate group onto ADP and we're going to make ATP. And so you don't actually make only 1, you make 2, right? And so now we have 3-phosphoglycerate because we removed one of those phosphate groups. And this is our first example of what we call substrate-level phosphorylation, where you take the phosphate from the substrate and then put it onto something else, right? So we've just now replaced the two ATPs that we generated, the two ATP that we used up in our investment phase, right? So we're net even right now. All right. Now, reaction 8, we've got a 3-carbon chain and we've got 1 phosphate on it. And our goal for reaction 8 is to make a compound that can be converted into a phosphoenol pyruvate. And this is because we need a second substrate phosphorylation event. We need to move that phosphate off. And so we need to create a molecule that will facilitate that. And so we have a mutase. A mutase is just going to kind of rearrange everything. And what you can see is you move the phosphate from the end to our middle carbon. And so it's just a phosphoryl shift. It is just getting the molecule in the correct orientation for reaction 9. Reaction 9 is the endolase. So the endolase functions through a dehydration reaction, right? So dehydration, we're gonna remove water, right? So we have like a hydrogen from here, a hydroxyl from here. We pull those off, we make water, and what are we left with? We're left with a double bond to the carbons and now 2 hydrogens on there. Now, what's great about this is, guess what? High energy, right? And we need these high energy molecules so that we can do those substrate level phosphorylation events. And so it makes it very easy to transfer our phosphoryl group from phosphoenol pyruvate to the ADP in the next reaction. So this is really similar to the Glyceraldehyde-3-phosphate Dehydrogenase catalyzed reaction in step 6, where we did that. So there are 2 places where we do a substrate level phosphorylation, and this is the second one. So this is catalyzed by pyruvate kinase. This is important because this is also irreversible, right? And so this again produces 2 ATP. So this is where our net 2 can come from, because at this point we had like broken even. But now we're going to get 2 more ATP, because how many of these do you actually have? You have 2, so you're going to make not 1 ATP, but 2. And you're not going to make 1 pyruvate, but you're going to make 2, right? And so this is that second substrate level phosphorylation event where we transfer the phosphoryl group from our substrate onto ADP to make ATP. And those are all of your steps inside of glycolysis, right? So you got to remember that this is the way that cells can make ATP if they can't use the enzyme ATP synthase, right? They can't go through that process. And so, and you remember that all of these steps inside of glycolysis have made our substrates negative. And so now those negative substrates are stuck inside of the cell, they can't leave, and we're able to get 2 net ATP molecules out of this. So it's important that you know where are you investing ATP, where are you getting ATP out, which reactions are reversible, which reactions are irreversible. At which stage are you controlling the overall flux of the entire pathway? There's one key step that we talked about, right? Where do we have those substrate level phosphorylation events, right? Those happen a couple of times. So understanding the key points of the glycolytic pathway are really, really important. Okay, and that is the end of section, was that section 3? All right, y'all have a great rest of your day.